Good evening, everyone. Welcome to the Professional Development Series for Virginia teachers. We're going to give folks a few moments to enter the webinar, and then we'll get started. Thank you for joining us, if anyone is out there. Let's give folks one more minute and then we can get started. In the meantime, I'll just give a couple of shout outs to the kind of sponsors of this webinar. So we have the Virginia Scientists in Educators Alliance, earthday.org, and the Chesapeake Bay Restoration Fund. So I will go ahead and kick us off, and as folks join in, they can get caught up on the webinar. We just have a couple of housekeeping to do in the beginning. I'll first introduce myself. I am new to the webinar series. My name is Matthew Arouk. I am the Director of Global Education with earthday.org. I started with the organization in January and up to this point have been unable to attend the webinars. I have two sons. They're aged 2.5 and almost 10 months this week, and their bedtime is 6:30 to 7:30 p.m. So I was unable to attend any of the earlier webinars. So here I am. Thank you for accommodating a new time so that I could join you. I'm pleased to introduce today's session. Our presenter is Amanda Laverty. Amanda is a budget analyst at NOAA and has a Master's of Science focused on biological oceanography from Old Dominion University. And she's going to talk to us about the Virginia Scientists and Educators Alliance, as well as share with us a lesson on plastics as a habitat for bacteria. But before we get started, just a couple of housekeeping items. The webinar is going to be recorded and live streamed, so you'll be able to kind of catch up with it after if you want to revisit what we have been talking about. There is a chat box for questions. We'll spend the last kind of 15 minutes or so of the webinar just revisiting those questions and answering them as best we can. If you are interested in closed captions, should be a closed captions icon at the bottom of your screen. Please go ahead and click that, and closed captions will be made available to you. And if you haven't already registered, please register to receive an email with the link to the recording and stay up to date on future webinars. After today's webinar, there'll be two more sessions. So nutrients and phytoplankton, and then on May 4th, a lesson on environmental justice. All of these activities, topics, and lessons are designed to increase awareness and knowledge about watersheds and the connection of ecological, economic, and social systems. We are specifically focused on the Chesapeake Bay watershed, and the goal here is to support all of you educators and all of your students to become active stewards of the Chesapeake Bay watershed. So the next one will be on April 4th. At the same time, 5 p.m. to 6 p.m. You can go ahead and register, and we'll send you the necessary registration links and updates. Before we get started, just a thank you again to the sponsor, the Chesapeake Bay Restoration Fund. They received their funds through the sale of the Friend of the Chesapeake Bay license plates. To learn more about how to purchase a Friend of the Chesapeake Bay license plate, you can go ahead and click a link in the webinar chat. And now I'll talk a little bit about earthday.org. So earthday.org, my home institution, our mission is to diversify, educate, and activate the world's largest environmental movement. So we work year-round, not just on Earth Day. We like to say it's not a day, it's a movement, with more than 150,000 partners around the world in more than 190 countries. And our goal is to drive action for the planet. So go ahead and take action with us. We have a number of education resources available to you on our Educator resources page. You can sign up your school as an Earth Day school. You can sign up to receive our Educator newsletter. You can sign up for an event on Earth Day. There are a number of different ways you can get involved with the organization. If you want to reach out to our program directly, you can email us at education@earthday.org. And then, so I just talked a little bit about these resources. Again, feel free to visit our website and find them. And then we currently have an ongoing policy campaign for climate and environmental literacy. And the mission here is similar to our goal for this webinar series, and it's to ensure that all students, all teachers, all learners have access to fully integrated climate and environmental education with a strong civic engagement component. So we give you the knowledge to know what's happening. We provide you with some of the skills that you need to take action, and then a roadmap for those actions. And here we have a 2-minute poll to find out a little bit about what you all know about plastic as habitats for bacteria. Says that I'm not allowed to vote because I'm a panelist. So with that, I'll go ahead and turn it over to our panelists. It's with great pleasure that I have the opportunity to introduce Amanda, who's going to be leading this lesson about plastic as a habitat for bacteria. Amanda, thank you so much for joining us. Audience members, thank you so much for being with us this evening. We know everyone is busy these days, and I'll turn it over to you. Thank you very much.
Thanks, Matthew. I will share my screen here and get started. Okay. All right. Well, good evening, everyone. Thanks so much for listening in, and a big thank you to earthday.org for the invitation to present today. I'm excited to do this. So the title of the lesson plan that I created is Plastic as a Habitat for Bacteria and Human Pathogens. Worked with Virginia C, which Matthew described, the Virginia Scientists and Educators Alliance, which is run by educators from the Chesapeake Bay National Estuarine Research Reserve in Virginia and the Virginia Institute of Marine Science, or VIMS, Marine Advisory Program. And in preparation for this discussion today, I saw that 32 students have created lesson plans with Virginia C since 2015. So there's lots of options there. And all of the lesson plans that went through this program were vetted by classroom teachers, and then a science fair like style open house was held each spring to introduce those lesson plans to educators. This one here invites 7th grade life science students to investigate marine plastic pollution as a habitat for bacteria and human pathogens. This lesson was developed in alignment with the 2010 Virginia Department of Education's Science Standards of Learning. And although it was geared towards 7th grade life science, it can definitely be modified and changed for, up through high school classes as well. So it can definitely be scaled however you might want to use it. And so using this lesson, students will examine the hazards of marine plastic pollution and learn about current science that's going on. And they'll also have the opportunity to use data to create graphs and discuss patterns. So a little bit about me. So I went to Old Dominion University for graduate school. And I studied plastic pollution and Vibrio Bacteria in particular. And this was sort of I was really interested in plastic pollution, and my advisor, Fred Dobbs, was working and has worked in his career, particularly focused on Vibrio Bacteria and ballast water, but mostly like microbial ecology. And there was a paper that came out right before I started graduate school with him that was titled Life in the Plastosphere. And it was about microbial communities on plastic marine debris out in the middle of the ocean. And I thought it was really interesting, and I thought it was a great way to meld both of our interests together. And so that's how we got started on the project. And Dr. Dobbs is continuing to work on plastic pollution at ODU, which is pretty great. And since graduating, I started working for NOAA, and that's where I am now. So what I'll do is I'll walk through the presentation that's available for download with this lesson plan. And then I've added a couple slides at the end to also walk through the lesson materials outside of the presentation itself as well. So this is the title slide for the presentation, and then sort of get into the nature of the problem, or that's how I've described it here. So basically showing, over time, just since 1950 to this shows to 2018, the global production of plastic and how it's pretty exponential. In less than a century, it's gone from invention to ubiquity, and there's some other lines here that show, how it's increased sort of along the same line as human population has. And then it also shows that laws introduced around plastic and plastic pollution really haven't come into, or haven't been super common until 2006 or so. And to further show the issue here with this enormous amount of plastic being produced, this graph shows some of what's landfilled and recycled. So the gray outline is plastic waste that's being landfilled, and the blue is recycled. So you can see not very much of plastic is being recycled, and so that won't get us out of the issue that we've found ourselves in terms of plastic waste and pollution. In 2018, only about 8.7% was recycled in the U.S. And so we can see that there's it's not great. So then also talking about like mismanaged plastic waste. So we have this waste problem. It's an issue generally. And with increasing populations, we have increasing amounts of garbage. And now with single-use items as a norm, it makes management even more difficult. There was a paper that was published in Science in 2015, Jambeck et al., who looked at 192 coastal countries to estimate the mass of mismanaged plastic waste across the world. And this is what came out of that paper. So, you know, in this slide, you can sort of explain the graph in colors. You know, the lighter colors indicate less share of a global total of this mismanaged plastic waste, where darker colors, darker countries are sharing a larger percentage of that global total. And then the grayed out countries just weren't incorporated in this study. So they calculate that 275 million metric tons of plastic was generated in these countries in 2010, and 4.8 to 12.7 million metric tons were entering the ocean every year. So you can sort of talk about how, you know, infrastructure in these different countries creates this mismanagement, and you know, talk about when we see like overflowing trash cans in a city, and how that trash, or, you know, plastic items can end up in the gutters and the sewers, and eventually into, you know, rivers, and then out into the ocean. Or when we see, you know, garbage trucks and trash coming out of them as they, you know, ride down the street. So, you know, various ways that the infrastructure is not necessarily the best, or could be improved, and certainly dependent upon money, you know, and whatnot in these different countries. But also, you know, looking at that past graph or chart, we can, you know, say that Indonesia and China, for example, like had much darker colors, so they are representing a larger total of the global mismanaged waste. But there's also, it's not exactly, it's not the whole story, I guess, right? So for example, America actually sends their plastic waste to different countries as well, and we're not the only one who does this. So it's not there's no simple solution or answer to these pretty big, large scale problems. And then just sort of talking about how, you know, how plastic enters the ocean in the first place. There's, you know, many ways, including like, you know, the fishing industry being out in the ocean and losing gear. But then there's also this large proportion of plastic waste that's entering the oceans from our rivers. And I think this is a good way to connect students, especially students who might not be in a coastal community or live right on the water, you know, like making that connection that the trash that ends up in the river near you will end up in the ocean, or can end up in the ocean eventually. But you can talk more about, you know, different modes of entry as well. And that, you know, this plastic, then it really stays around for a long time in the environment. And most plastic items never fully disappear. They just break down more and more. And so here can ask students, you know, what they can identify in these pictures, and like what looks familiar to them. So like in the bottom right picture, you can see there's laundry jugs and cans and bottles. In the middle picture, there's a plastic bag there, which can resemble a jellyfish, which is, you know, pretty devastating for sea turtles and other animals that feed on jellyfish. Up at the top, you see a lot of ropes, which is, you know, that like fishing gear waste. And then you can talk about, you know, how these different types of everyday items end up in the ocean, and then talk about the breakdown of them. So in the left photo there, these are mostly microplastics that are being shown, and you know, defining microplastics are just really small pieces of plastic that are typically less than 5 millimeters in size. And you know, much of the plastic in the ocean is small because it's being broken down by, wind and wave action and the sunlight hitting it. And so either it's being broken down by these larger pieces of plastic materials, or they start out small. So going a little farther into microplastics, there's a difference between primary and secondary microplastics. So primary microplastics are plastics that are manufactured products that are used in everyday, household items like facial cleansers. So you can see in the bottom right picture that there, are these tiny little microbeads that are in these various cosmetics. Or in the toothpaste there on the left, like those tiny little dark blue, pieces or flecks there are actually microplastics. And microplastics in these kinds of items were banned in the United States in 2018, but not necessarily in across the world, in the US though. And then there are plastic pellets, resin pellets up on the left. And so these are considered primary microplastics as well. So they're, small, like cylinder shaped plastic pieces that are typically created so that you can remelt them and mold them into new, larger products. And it just makes it a lot easier to ship these around. And they certainly, at least to me, look like they could be something naturally found in the ocean environment, like, fish eggs or something like that. And so, that creates a problem for these, creatures who are trying to feed on things like that. So those are all primary types of microplastics. And then we have secondary microplastics, which are on the top right photo, this person holding these pieces of plastic. So these are, pieces that have been broken off larger plastic objects, which happens a lot. So those are secondary. And then sort of get into, what is the Great Pacific Garbage Patch? I think a lot of folks have heard about the Great Pacific Garbage Patch, and it seems that the way that we've publicized it in the media has been a little bit maybe misleading. And so, maybe asking the students what they've heard about it or what they know about it, and then explaining really what it is. So I think a lot of the way that we've been messaged is that it's this, island of trash. And I think it leads people to believe that there's this island that you can walk on, or something, like there's, a ton of trash out there on the surface that you can really see. And that's not necessarily the case. In fact, these garbage patches, actually, it's all throughout the water column, all the way down to the seafloor. And, just getting into this, so this garbage patches are just basically large areas of the ocean where, litter accumulates. And they're formed by ocean currents called gyres, and, explaining them as like whirlpools that pull objects in. And then, we form this patch essentially. And the most famous one is the Great Pacific Garbage Patch, and it's located in the North Pacific Gyre, which is between Hawaii and California. And you can see that this was some water collected in the Great Pacific Garbage Patch. And if you look hard, you can see there are, different colors here, like blue and yellow and red. And so these are all tiny pieces of microplastic. And it's been noted that in the North Pacific, in that Pacific garbage patch, that plastic actually outweighs surface zooplankton 6-to-1, which is pretty incredible. And then further, it's not just actually in the North or in the Pacific, it's actually in all of the major oceans. There are actually 5 gyres, and trash accumulates in every major gyre. And then there's a little video here that's linked at the bottom that it's about 3 minutes or so that you can watch to see how much plastic is actually in the oceans. And then here, maybe brainstorming with the students why plastic is a problem. You've already given them some reasons, but There are many. So, on the beach, there's aesthetic issues. It can hurt the economy by keeping tourists away from beaches. Nobody wants to, go to a dirty beach. And also, impacts the animals that are living on the beach as well. And then there's entanglement issues, ingestion issues by macroplastics and microplastics. Other kinds of entanglement on coral reefs, for example, and then navigational issues can be a problem. And then potential transport of invasive species or species that shouldn't be in other parts of the ocean. Like these are filter feeders that are attached to these bottles. And then ghost fishing is a major issue for the economy as well, and animals. And so speaking of the transport issue, that's how we get into the potential of bacteria living on plastics. And so this is a good point to talk about the microbial biofilm life cycle and explain biofilms and how they protect bacteria, and that bacteria live on many surfaces. And they utilize these biofilms because they create living conditions that protect bacteria from the environment. And they are found on plastic in the oceans, and plastic serves as this habitat for bacteria. And plastic is a long-lasting substrate, versus something that might break down in the ocean, something organic, and that it can move with the ocean current. So there's that potential for transport of bacteria as well. So then this gets a little bit closer into the project itself. So talking about Vibrio bacteria particularly. So Vibrio is a genus of gram-negative bacteria, and they have this, rod shaped to them, and are able to move around. And there's several species of them that can cause foodborne infection, which is usually associated with eating undercooked seafood. And they're typically found in saltwater, and they're very dangerous when pathogenic. And then the transportation or ingestion of these bacteria could be impactful for human health and marine animals. And so there's some definition here in terms of what a pathogen is, and these, and also their temperature and salinity dependent. So they thrive in warm temperatures and in high salinity. And then, talking about the scientific method. So, we use the scientific method to investigate and acquire new knowledge. And part of that is coming up with a hypothesis or an idea or explanation that you test through study and experimentation. And then, we do this in all of these different fields. And we all, scientists in all of these different fields ask different questions and perform different tests, but they use this core method to investigate what their questions are. So for this project, the hypothesis was, if plastics remain in the marine environment for long periods of time, then plastics may be a vector for bacteria and human pathogens. So this was the project study area. So the lower Chesapeake Bay, I put a little square there around the bottom to show that this next photo on the right is blown up from that area. And so the star represents the Elizabeth River, where I worked with microplastics particularly, and then the circle is in the Lafayette River, where I did some colonization experiments, and that's mostly what we talk about in the activity here. So we set up 2 experiments on a local dock in the Lafayette River, and we hung these different kinds of plastic types, the picture up there on the far left, from a floating dock, and we sampled the biofilms on each of those different kinds of plastics, and we did it over time with replication. So we looked at days 1, 2, 4, 8, and 16. And so you can see that the biofilm was removed on that right hand middle picture from the left, where was not removed. So that was right after we pulled it out of the water. So biofilms were scraped off a section of the plastics, and I was careful to do just like 2 centimeters. So there was a way to normalize at the end, which is something that the students will have to do, and I'll explain. But and then, you know, the bacterial solution essentially was filtered onto chrome agar, which is a selective growth medium for Vibrio bacteria. So you can see that these colored colonies here on this plate, it's actually on chrome agar, they grow in different colors based on specific enzymes that are produced from different species of Vibrio bacteria. So like the purple, for example, is typically Vibrio parahemolyticus, and that's not always the case. There's some, you know, level of certainty with these, but it gives you an idea of what you expect it to be, and it should be confirmed through PCR. But it's fairly cool medium to use. And so these colonies can be counted and recorded as colony forming units, or CFUs, after 24 hours of growth time. And yeah, then we'll normalize to the area scraped and calculate how many CFUs per area. And then there were water samples collected in tandem to compare with the plastic colonization over time. And so this, at this point, it's, you know, you can define colony and colony forming unit and chrome agar and talk about that a bit. And then it gets into the classroom activity overview. So essentially students will count colonies on plates for 2 different substrates. So for polypropylene and low density polyethylene, so 2 different types of plastics. And water in replicates of 3 and over time. So there's the total plates to be counted would be 39, excuse me. And, you know, here is a good point to explain like reproducibility and replicates and why those kinds of things are important in science. And then, yeah, so students will count these different CFUs and then record them and graph them. And so the picture here on the right is what those photos look like. So there's a whole document that you can download with the photos labeled for each of the days and the different types of substrate. And so, yeah, so we can get into some graphing. Yeah, so then I didn't get into this with the normalization. So for the area, for the plastics, you would normalize by 2 centimeters squared, and then for the volume by 2 milliliters. So they'll just want to divide their CFUs by those. So then I put a little bit of a, you know, graphing overview here, and you can consider more or less review of graphing, you know, dependent upon your students and whatever's necessary. But I just, you know, sort of explain what, you know, independent variable and dependent variable and what that graph setup looks like. And then we can get into an example of that, and whether you want to have the students try it and then show them the result or do it together. But this is a good example where we have water temperature and time. So we can talk about what's the independent variable, what's the dependent variable, and then how that would look. And I have this temperature and time data in one of the handouts as well. So you can use that information to generate this graph and have a fairly simple graph to start out with, because the graph with the plastics and water CFUs get a little bit more complicated. And so this just indicates the end of that presentation. So then, so this is what the student handouts look like. I just, you know, put some, or what some of them look like. I just put some tables here that they can use to record their numbers. And then there's an answer key with everything. And here's an example graph of sort of what that would eventually look like with all of the 39 plates counted. So this is over time. And colony forming units per centimeter squared for the plastics or per milliliter for the water is on the y-axis. So you can see the blue and the red are those different types of plastic and what those colonies or how many colonies over time we saw. And then same for the water in green there. And so the difference here is that, you know, the plastics increased as the water decreased. And that's because the temperature got colder as this experiment went on. And so, you know, the Vibrio weren't really able to thrive as well in colder temperatures. They really like warmer waters. And then, you know, on the plastics, they were sort of protected by that biofilm. And so they were able to grow a lot more of them and still, you know, maintain that growth over time. And then these are some of the discussion questions that are in the handout as well. So just talking about, you know, what are the trends that we see for these colonies on plastics over time? Are both plastic substrates showing the same trend? You know, what trend do we see for the water? And then how do we compare that to water temperature and time? And are they related? And then, you know, finally, was our hypothesis correct? So could plastic pollution be harboring and possibly transporting bacteria in the marine environment? So thank you so much for your time. I hope you consider using this lesson in your classrooms. And if you have any feedback, I would be excited to hear it. I put my contact information here, and I also took a screenshot of where these lesson plans can be found. And I put the URL at the bottom there. So please, don't hesitate to reach out if you have any questions. Thank you.
All right, thank you so much, Amanda. We have a couple of questions from the audience. The first one is a practical question, right? So are scientists developing ways to remove some of these microplastics from the oceans, or are we helpless as a society once plastics get to that size and are in that marine environment?
Yeah, that's a great question. I think scientists are definitely trying, working on different kinds of methods to get plastics, generally plastics out, and microplastics. I think it does present a pretty big problem when they get smaller, because they're suspended through the water column, and it's hard to filter those out and not, you know, interact with animals too. And so I think we are looking at ways. I think there's a lot more studies. There's a lot of, well, I guess there's a lot of like prevention kind of studies that are going on too. Like how do we prevent plastics from getting there in the first place? What kind of, like, you know, behavioral change can we create to try to minimize that initially? But yeah.
That's a good segue to the next question that came from Twitter. So thank you, Twitter participants. And that's, what actions can individuals or communities take to reduce plastic pollution before we get to that problem of runoff into the ocean?
That's a great question. I mean, I guess there are lots of things we can do in terms of reducing our plastic consumption, recycling as much as we can, although, you know, like we talked about initially, that's definitely not going to get us out of the issue, but yeah, trying to reduce and try to buy in bulk, and, you know, just do your part in terms of consumption. And then, you know, also participating in these kinds of like beach cleanups and things. It looks like you guys do some of those. So earthday.org, like participating with those, or the International Coastal Cleanup does a big one across the world. So stuff like that is a great way to give back and be a part of some change.
And then to sort of take a different pathway, this is an educator question. I'm just curious, you know, what age group do you imagine this lesson being most appropriate for? Is there specialized equipment that folks would need to implement it, to do that kind of analysis? And then would you be able to modify it for older or younger age groups, depending on where the teacher is or where the students are at.
Yeah, I think so. So I tailored it to 7th grade standards of learning, but I think it's applicable to even, through high school as well. And yeah, I think, you can, for the graphing portion, I think you can probably graph on paper. Or you could do, in Excel. So depending on the resources that different schools have, different teachers have, they can do either or. I think the only thing you actually need would be to print the photos of the images, and to have the PowerPoint presented. But yeah, I think, the PowerPoint is there and can be modified however as well. So I think it's fairly open to, whatever the teacher is looking for.
Great. Thank you. And then I have a question about the graph that was in the slideshow earlier, and it was really interesting to see which countries were shaded darker than others. And I'm just curious if that's a per capita shading, or if that's kind of gross plastic consumption and use as a management graph.
Yeah, that's a good question. I'd have to probably go back and review exactly the details of it, but I think what the graph that I had specific was like a percentage of the total mismanaged waste. So the waste that we're seeing that's potential to enter the ocean, based on like different kinds of infrastructure and those kinds of things. Yeah, I'd have to go back and review exactly the paper though.
And then a final question for you, and this is kind of looking at a future webinar. And again, this is a Matt question. What are the implications here for kind of environmental justice? That's going to be the last topic that we explore in the webinar series. And I'm just curious your take on kind of the implications for equity and justice as it relates to microplastics and pollution more broadly.
That's a great question. I mean, do you have thoughts on that? I guess, yeah.
Well, let me try to make it a little more. So you worked with these kind of 2 river areas at the south part of the bay. How are those communities dealing with that issue? Were they aware of the issue and its presence? Were they aware of the kind of sources of pollution, maybe upstream, that were affecting their communities, fisheries? I mean, was there any kind of conversation around how to account for this issue inside of these communities, despite the fact that some of the resolution might need to take place further afield?
Yeah, I mean, in my study, there wasn't a whole lot of that went on, but I think, yeah, I think it's a great point and something that could be looked at further. I mean, I could imagine that, some communities are more impacted than others in terms of plastic waste and trash that's littering the area. And I could see, maybe focused cleanups in different areas versus others, and how that could be an equity problem, like access to those kinds of spaces. Yeah, and then in terms of like, I'm thinking for my project, I focused on like human health particularly, and so I'm not exactly sure how this would fit into environmental justice, but it's certainly a problem for human health in terms of like oysters or other kind of filter feeders potentially that are filtering in these microplastics that might have bacteria, like a lot of bacteria associated, and then we go and we eat these oysters, we take them out of the Chesapeake and eat them raw, and how that could maybe increase the amount of those Vibrios that we're coming in contact with potentially, and whether or not they're pathogenic. I think there's more research that needs to be done with that, but I think there are definitely potential for human health issues there. So
yeah, and probably certainly affecting the livelihoods of folks in those communities who rely on oyster sales or oyster consumption for either their
Yep.
food security or for their tourism or for their hospitality or for their culture, right? Some of these communities. So
absolutely.
Great. Well, do you have any final thoughts that you'd like to throw out before we wrap things up?
I don't think I do. Thank you so much for the great questions and for inviting me. This was great.
All right. Well, thank you very much, Amanda, and thank you to the audience in person and in Twitter and wherever else you may be viewing our webinar. Thank you so much. If you're interested, please sign up for the series for updates. There you'll find resources, recordings, everything from past and future webinars. And like today, we'll have another lesson that you can bring home to your students on April 4th. Instead of talking about plastics and microplastics, the next webinar will be focused on nutrients and phytoplankton. So thank you again, Amanda, for the wonderful presentation. And thank you everyone for joining us this afternoon. We look forward to seeing everyone again soon. Thank you and take care on behalf of earthday.org.